It's a huge pleasure to be here with you today. And I think that all I am is just someone who's been dedicated to making a difference for cancer patients in different ways. And I think in watching your scrolling zebras, one of the things was, make a difference. One way we as patients can make a difference is to participate in clinical trials. But it's hard to feel confident that you're not the zebra striped laboratory rat unless you understand what it actually means to be a clinical trial. So let's delve into this, starting with the fact that I don't have any disclosures. It is true when you work for the government, you need a hall pass to go to the bathroom, so it's pretty hard to have other things that are conflicts. The big thing is that in order to make progress, we have to do clinical trials. And in order to do clinical trials, we have to have patients who trust in the system and understand it to participate. Only 3 or 4% of patients with cancer actually go on clinical trials, where it's estimated even a third or more of patients could be eligible at a given time. In contrast, unfortunately, in childhood cancer, it's about 95% participation. That may be why they make as much progress as they do. Unfortunately for us in the field of neuroendocrine neoplasms and neuroendocrine cancers, for reasons that we don't fully understand, the incidence is on the rise. Maybe it's because with all these CAT scans and things that people are doing, we're picking them up earlier before they're symptomatic or metastatic. You've probably heard on the radio, oh, come get your cancer finding CAT scan. Well, you have no reason for a CAT scan, but you go in and get it, and sometimes they find something. But the fact that neuroendocrine cancers are on the rise over these past decades is certainly of concern and increases the importance of us making progress. And so we really do need more clinical trials. As you can see, from 1980 to 2020, there was a desert of advances for decades. But now, in the more recent time, we are seeing some progress with lanreotide, telistostat, everolimus. By the way, if you wonder why they have all these horrible names, it's because they want to try and trip us up. But all of these things, including now PRRT, they're recent progress, but we have to understand how best to use them. So, what do clinical trials do? They sit in a simple way, they test what we think to be good and safe ideas that should make a difference for the patients we serve. So in English, a clinical trial, which is down at the bottom in blue, is a research study in which people participate. Sometimes there are more than one arm of a health-related intervention that is evaluating something that's predefined. So when should you participate in a trial? Anytime. It's important to understand many cancers have trials for you at the time of diagnosis, at the time you may require treatment, or at the time you may require treatment for recurrent or progressive disease. But why a clinical trial? They allow us to progressively assess clinical interventions, whether it's drugs, radiation, imaging, surgery, or other things, but it requires that we formalize what we want to find out, how we're going to do it, how few patients we need to get an answer we can believe, and in a way where we can maximize patient safety. In short, we want the return on your investment as a patient to be the very greatest, no matter what trial you're in. These trials provide the evidence that change the practice that the doctors use, whether they're in private practice or in ivory towers. So a clinical trial is important also because, keep in mind, if we're doing something experimental, we've got to watch carefully. So the care and oversight may actually be greater because it's a requirement for patient monitoring and data collection. So there's something good about clinical trials, not just the fact that you're providing yourself to help make a difference for others. Who performs trials? Well, any investigator can theoretically perform a trial, But there are regulatory and safety legal guidelines. There are hurdles of education that need to be passed so that you can be trusted as an investigator, so that your patients understand that when you say that you're running a clinical trial, they can have confidence. So you as patients can have confidence in your doctors, like Dr. Chauhan, who has studied long and hard and gone extra steps to be able to understand how to be an investigator of clinical trials. The National Institutes of Health use your tax dollars to fund grants and contracts across many disciplines of medicine, not just oncology. And also investigators who are at the NIH out here in Maryland, Washington, can run trials. We have a hospital called the NIH Clinical Center, which can take patients from all over the country if they're eligible for a particular trial in which we are involved. But also, industry runs trials. You hear them on the TV all the time, touting their results and trying to get you to take whatever drug that they're showing you. So who participates in clinical trials? Anybody who's interested, who fulfills the eligibility criteria. Those criteria are determined so that we can optimize your safety as a patient. And there are some trials where they use normal volunteers, which in those cases are healthy people who are volunteering to take an agent or undergo an intervention for the purpose of providing scientific guidance. So who should participate? Well, my feeling is everybody. So now I understand that earlier today and yesterday you had a lot of questions like, what is a phase 1 trial? So let's delve into it. What is a clinical trial phase? Well, they are different progressive steps where we learn different elements in a logical order, because you don't want to do something because it works if it's not safe. So safety and the right dose and schedule are early, and those are the subject of phase 1 and early phase 2 trials. Demonstrating activity in a more similar group of patients is what we see in phase 2. And in phase 3, it's a duel between the standard of care and the new idea to see who wins or if they're equivalent. So in phase 1, 2, and 3, we have different objectives, but across the board, we're looking for safety and clinical benefit. So again, in phase 1, we're trying to see if we can safely dose the drug in a way that it's, or agent, in the way it's most beneficial. And oftentimes we'll invite a large variety of cancer types, but not always, because we might want to be looking for that needle in the haystack that says this is a hint in this direction or that. But our specific focus is safety. While we watch for individual benefit, that's not one of our predetermined statistical endpoints to which we're held. That comes up as we move towards phase 2. Again, a more common group of patients, maybe all neuroendocrine patients. A consistent treatment approach, dose and schedule, timing. Looking for the beginning of activity information. And sometimes we can randomize it because sometimes we're adding something to an existing standard. So we need to determine, can we do that safely? Does it make more bang for the buck? And then in phase 3, that's where we're really going to say, this is going to change how we do business. Now, sometimes with rare cancers, like some neuroendocrine cancers, we only go as far as phase 2, but we adjust that phase 2 so there's a level of stringency and enough patient participation that we can be confident when we tell you that this is a direction we recommend. So this is all about building evidence to give you the best recommendations as patients. In phase 3, occasionally you will see something that's blinded or placebo-controlled, and that's to avoid purposeful or non-purposeful bias. Sometimes, I'll say, I'm not sure this patient can really handle all these extra things. But if I don't know what arm, then I have to either say yes or no at the front instead of biasing going in. So these are the stringency steps that we use to try and build the best information, the best evidence for treatment care. However, it is sometimes daunting, and I love looking for good medical cartoons. This one, for those of you in the back who might not be able to read it, after reading all about cancer treatments, trials, phases, and stages, I've decided to quit reading, which is something that I sometimes think about too. What goes into designing a clinical trial? Well, there needs to be science to justify it, not just because it's there or because somebody wakes up in the middle of the night with an epiphany. There needs to be data to support it. We're not going to subject patients to something that's not founded. The cartoon is we found a mass. The good news, we have weapons of mass destruction, but we want to be sure that our weapons do mass destruct before we apply them. We want to have a clinically meaningful target and difference. We don't want to do Coke versus Pepsi, and we don't want to do Tweedledum versus Tweedledee. We want to be making a big step because people are volunteering their time, their life, and their safety to be on a trial. It has to be interesting. The cartoon is essentially people vote with their feet. If it's interesting, people want to do it. If it's not, it's never going to accrue. But it has to be feasible. If it requires a huge amount of time, if it requires a large handful of pills, if it requires flying across the world, those are not feasibilities. And of course, the cartoon here is one convenient dose, but it's the size of the person. So these sound logical, but we have to put each of these steps in as we decide how to go forward. The important purpose of all of this is that there are exciting new directions in neuroendocrine tumor and cancer treatment. They're developing as we speak, and it's a great time if you have to, unfortunately, have this disease. Lutathera, lutetium dotatate, PRRT, use whatever name you want. We all agree, huge advance, big difference, but on the far right, you can see a banana effect where the curves come together. So it didn't change the cure, didn't change the number of people who were cured. If that were the case, the red line would have stayed above the blue line. That tells us that we've made a big difference, but we have to improve upon it. Capecibine with temozolomide, work done by Pam Kuntz and ECOG Akron for patients with pancreatic neuroendocrine tumors, made a difference in time before tumor came back, but it didn't change where that curve plateaued. An exciting advance, but we have to learn right time to use it for the right patient to make the biggest difference. So exciting directions right now that are going on in the federally supported world are building on capecitabine temozolomide for pancreatic neuroendocrine cancers by saying, what if we push it up really early for the subset of patients whose disease has the highest risk of returning? So giving it in adjuvant, can we delay the time to progression and maybe also push off and improve survival? Can we even make that surgery effect last longer? What about PRRT? As I just showed you, we need to understand the best way to use it and maybe even how to enhance it. So we want to know in the first approach, is it better than everolimus for patients who have lung neuroendocrine cancers? Because everolimus is something you take every day, is a little bit on the rough side after a while. And while it had some benefit, it's not huge. So can we help patients with pulmonary neuroendocrine cancers? On the other side, can we modify PRRT, as Dr. Chauhan has been doing, to make it better and maybe ultimately separate those overall survival curves? So if we use a new agent, one of the new targeted agents that may make the PRRT, the Lutathera, lutetium dotatate, better, will that make a difference in the long run? And then can we build on what we learned in the 90s and the noughts about drugs that inhibit blood vessel development, like sunitinib and bevacizumab? Can we approach them better? And we have a trial looking at a drug called cabozantinib. Would that be something that's both tolerable and active? So all of these things we want to understand, and we want to do it specifically for patients with neuroendocrine cancers. We don't want to have to extrapolate from another cancer because that's guesswork. So we've made a lot of progress, but right now we have more space to advance. So this is kind of busy, complicated, but I'm going to give you the hints to it. Vertically by color is the kind of cancer: pancreatic neuroendocrine tumor, other GI neuroendocrine tumors in blue, lung neuroendocrine in gold, the neuroendocrine cancers, the really aggressive ones, in red, and then paraganglioma pheochromocytomas over on the gray side. And then if we go across that first thin, that's when we give treatment right after surgery. And we have the study where we're looking, as I told you a moment ago, about capecibine temozolomide versus observation. And then in recurrent, locally advanced or metastatic neuroendocrine tumors and cancers, you can see there's a wide variety of different things going on, from cabozantinib as a single agent versus placebo to PRRT versus capecibine temozolomide, modifying PRRT by adding new drugs. That's in the blue set. In the gold set, the one I mentioned about PRRT versus everolimus. So these are all studies that are being funded and executed nationally through the NCI funded networks in a way to try and improve where we're going and the opportunities that we're creating with evidence to support your treatment that I hope you won't need in the future. So as we go into a period for questions, my last cartoon for you is the mouse. I go home today. They cured me using this new miracle drug. I'm afraid it'll be years before it's approved in humans. And that's the reason we need to do our trials and make sure we get the word out and encourage everybody to participate in trials, because that's how we move the bar forward and that's how we make a difference. And I want to say thank you for the opportunity on behalf of the NCI and all of my colleagues, many of whom are there, because their dedication is what helps make a difference for your care. Thank you very much.